Alright, so in this video, we are going to go over mitosis. So at this point, we have already gone through interphase, so we have already replicated our chromosomes. And what you'll notice is we have our centromere, and then we have our genetic information on our chromosomes. So in the first part of mitosis, we have prophase. Now what happens in prophase is the nucleolus disappears, the nuclear envelope disappears, and our chromosomes condense into chromatids. So these would look more like chromatids. So for example, your chromatin would be like a long piece of string, and then a little protein comes along and basically winds it up so it's nice, neat, and compact. Something like this, which we'll say transitions to this top part. So the reason why we want to condense our chromosomes is so they can be neatly organized. If I say, for example, this is what it would look like. Imagine trying to separate this and evenly make sure that each gets a copy. That would be insane. That would be chaotic. So what the cell does to be like, ha ha ha, I'm super organized, is it condenses the chromatin into chromatid. So we have completed prophase. Now in metaphase, what happens is the chromosomes line up along the center. Oopsies, one's falling apart. So they line up along the center in the middle, because metaphase, think of the middle, and then you have basically these spindle fibers that attach to the centromere. So they will attach on all sides, and this is what will allow us to separate our sister chromatids, because if you remember, this is an exact copy of this. So basically what will happen is spindle fibers will attach to every centromere. So it'll look like this, except it'll be a little nicer and neater. And this will happen to every chromosome. Then in anaphase, what happens, I'm going to move these out of the way so I can clearly show you. In anaphase, what happens is the spindle fibers will pull apart and pull a sister chromatid to each side. So then each side will have a chromatid of that copy of the chromosome. Boop. Pulls it apart. It's a little better than my string and some magnets. And so this is what your anaphase will look like. And then in telophase, what happens is these spindle fibers will disappear. Whoopsies. Our spindle fibers will disappear. Our nucleolus will start reforming, our nuclear envelope will start reforming, and then we will end up with almost two cells. It's not until cytokinesis where we actually split into two cells, but let's not forget about those chromosomes that I had before. So they'd split, 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 and split. And so you end up with, now each of your daughter cells has 2, 4, 6 chromosomes. This one also has 2, 4, 6 chromosomes. So keep in mind, in mitosis, our initial number of chromosomes will be the same in the initial cell, and then each daughter will have that same number of chromosomes. If you remember, the way we started was something like this. But keep in mind, at this stage in our initial cell, it had already been through the cell cycle. So if you think about it, if we were just in G1, what our original cell would actually look like would be something like this. And if you notice, there are 6 chromosomes, and our daughter cells ended in 6 chromosomes each. So that is what mitosis looks like.